shorter. So this is really the cardiometabolic disease space, the PCSK9, which is very interesting to many people because we're talking about what's after statin. People still have inertia. The physicians still prescribe statins as the kind of preventive therapy for high cholesterol or hypercholesterolemia. And this is very successful. So far, this is probably very few examples of preventive care finally got bought into the general health authority. And so the insurance company typically can cover the statins for many people. But now we're entering the era that is statin enough? Is just taking statin enough? Lower the bad cholesterol enough? So that's a very important question. So that's why PCSK9 is a very fascinating target. It's a very short history. It's only discovered in 2003 through very elegant genetic analysis that when people study familial hypercholesterolemia, the usual suspect is the LDL receptor. That's the most obvious one. And the second most common genetic linkage is apoprotein B. And apoprotein B, as you know, is the main lipoprotein on the kind of LDL in the low density lipoprotein. That's why that's not surprising. LDL and ApoB, thanks to Brown and Goldstein's Nobel Prize winning discovery, so that's a very important pathway for the low density cholesterol to do its damage. But it turns out by studying the familial hypercholesterolemia even deeper in 2003, these researchers in France was able to discover a third gene, which is very high interesting link. It's called PCSK9. At that time, the name was not PCSK9. Luckily, when they identified the autosomal dominant mutations, they identified two mutations. One is the serine 127 to arginine. The other one is the phenylalanine 216 to leucine. These two mutations turn out to be very intriguing because they are autosomal dominant, meaning they apparently seem to have some gain of function. The gain of function meaning the loss of the gain of function turned out to be making the cholesterolemia worse because it boosting up the PCSK9 function. So later they found out this gene or the protein sequence already identified as NAC1. NAC1 is neuronal apoptosis regulatory gene. So that one turned out to be in the family so-called PC, stand for protein convertase. As people know, there's many important family, the PCs. Those proteases process the many hormones post-translationally and leading to many famous numbers like PC1, PC2, and PC3, and is also known as furin. And it turns out that the first 7 members in this family are the canonical protease cleaving after arginine or lysine, very similar to trypsin. So it's cleaving after the basic residue. But the 8th member is called MBTPS1, turned out to be different from the first 7 member, cleaving after LVI, then followed by X, any amino acid. So this new NAC1 belongs to the 9th family member in this family. The yeast ortholog is called caxin, and the bacterial ortholog is called satylicin, stemming from Bacillus subtilis. So it turns out that the PCSK9, the 9th member, turned out to be protein kinase K-like, but interestingly, it's cleaving at the different residue. It's cleaving at after a glutamine Q. So that's a very interesting finding. So first, you have some gain of function, but they're still not 100% sure it's gain of function until 2005 and 2006, they discovered there's loss of function. The loss of function turned out to be lower the cholesterol. So that confirmed the 2003 discovery. The gain of function increased the cholesterol. So then when you study this PCSK9 protein function, turns out to be very different from the other family member. Like I like to always fascinated by this example is the GLP-1 and GLP-2. People are very famous now known for GLP-1. It turns out for GLP-1 to be processed has to be differently in pancreas and intestine. When this pre-pro-glucagon polypeptide, they contain both glucagon, GLP-1, and GLP-2 in the pre-pro form. But after cleaving off the signal peptide, you have this pro-glucagon undergoes differential post-translational processing. In the pancreas, the protease PC2 will cleave into a functional glucagon, but the GLP-1 and GLP-2 fusion protein is not functional. That's why you won't get GLP-1 from the pancreas. On the other hand, in the intestinal cell, they have a different protease called PC1, which cleave differently and leading to the glucagon GIPP lumped together as inactive form, and GLP-1 and GLP-2 are functional. So these are the traditional famous members in the PC family, but turns out PCSK9 is very different. PCSK9 only cleaves itself. It does not cleave any other protein. So after the signal peptide cleave off, it cleaves itself at the pro-domain boundary. So once the interesting is that once the pro-domain got cleaved off, it become a blocker. It become a chaperone. It will blocking the catalytic activity of this protease. So the mature form is a enzymatically inactive serine protease. So that's an evolutionary kind of very interesting example that the ninth family member no longer function as a protease. And this chaperone complex will shuttle this whole mature form into secretion. It will be secreted out of the cell. And then once it's outside the cell, it will function differently. So that's the kind of fascinating part of the ninth member is no longer a protease. It turns out that once it's getting secreted out of the cell, because the pro-domain blocking its catalytic activity, many years of research then found out it will bind to the LDL receptor. It will bind to the LDL receptor, and this complex will degrade the LDL receptor so that it's no longer available to clean up the LDL. So this definitely, you can see when it's functional, it will leading to the increase of the cholesterol, circulating cholesterol in LDL in the blood. That's why if you can design a monoclonal antibody, because it's coming from genetics, two companies are very focused, really emphasize the importance of genetics in the industry is Amgen and Regeneron. So not surprisingly, both companies acting very quick on the strong genetic evidence. And of course, both Amgen and Regeneron are also very strong in developing monoclonal antibodies. So they are the first two made the finish line. And it turns out the other three followers, like Pfizer, Lilly, and Roche, they're a little bit slower. It turned out to be once these two get onto the market, those three players, the Pfizer is because they're not as durable as Repatha and the Praluent. Repatha from Amgen, Praluent from Regeneron, and Pfizer is just not as efficacious, whereas Lilly and Roche strategically decide they don't want to be the third antibody against the same target on the market because they had to compete with statin anyway. So end up, you only have two monoclonal drugs approved for the PCSK family, but they look very efficacious. They can lower cholesterol, LDL-C, much lower than the statin. They can reduce like 55% reduction of LDL. So, but in terms of market acceptance, because it's like Jun said in the other example, they are auto-injector. The best formulation so far, subcutaneous auto-injector for every 2 weeks. And initially the price was not friendly, but in recent years, the price started to drop. And so now the kind of affordability of health economics is no longer a problem. But the physicians still have this inertia of automatically prescribe statins over this new class of PCSK9, because the argument is that you want to lower LDL-C further for those people with higher risk. But for people generally already on statin, it's very difficult to convince them to switch from an oral drug, daily oral drug, to an antibody injection every 2 weeks. So that's one of the kind of difficulty, although they sound reasonable, but they can never meet the original expectation in terms of competing with statin. Then many years later, Alnylam was able to enter this space using the siRNA approach, which is very promising because siRNA not only can take its action extracellularly, blocking the PCSK9, it actually go inside the cell. So once you go inside the cell and degrade the mRNA, so essentially it's just acting both the intracellularly as well as preventing the protein from going to the outside because they're degraded, the mRNA degraded. So on the other hand, monoclonal antibody cannot blocking the other pathway, which is the PCSK9 protein can bind to LDL before getting secreted in the trans-Golgi network. It can still find the LDL receptor and degrade it. So theoretically speaking, RNAi kind of silencing should work better because it can act both intracellularly as well as extracellularly. But the real data turns out it's not the case. It's interesting that probably the extracellular mechanism is more important. So if you just compare LDL lowering for the antibody compared to Leqvio, the siRNA, numerically, the antibody is even better. But of course, the main advantage for Leqvio siRNA is it can be durable because it's not occupancy-based pharmacology. All the antibody working on as occupancy-based pharmacology. But siRNA is more like a intracellular gene silencing pharmacology. It can be durable. So once the guide strand of siRNA getting loaded into the risk, it can be very durable. That's why after first 2 doses, Leqvio can only need to be injected every 6 months. So you only need 2 injections a year. But there's a very important caveat. That injection is not auto-injector. It's sub-Q injection, but it needs physician to perform that injection. So that's still not very convenient, meaning the patient still need to see the physician twice a year, but of course it's once every 6 months. And also the health economics, so far the price with Alnylam and Novartis has not dropped high enough. So that means Repatha still have some kind of competitive advantage. Many insurance companies still will give the prior authorization for Repatha, and many insurance companies don't even need the prior authorization, whereas the Leqvio, 100% you need prior authorization, and the prior authorization sometimes can be denied, even it's granted. The price out-of-pocket expense for Leqvio still not as friendly as Repatha and those antibodies. So that's why it's an interesting competitive landscape down the road. But Merck's oral PCSK9 inhibitor really becomes potentially a game changer in all this competitive landscape, because for oral drug daily, like statin, it will be very advantageous because the compliance will be huge. But to make an oral drug works for PCSK9 is not easy because it's not a protease, it's an enzymatic inactive protease. It's not like a traditional protease. And you need a very innovative medicinal chemistry to make PCSK9 druggable. So it takes Merck's many years. And it was eventually they succeeded. And Dr. Hongyu Zhao talked about this oral micropeptide back in March, and the video is on the YouTube. If you search BioSpark group, you can look at Hongyu's deep dive into how to develop oral micropeptide. It's a very fascinating story. But to make a long story short, Merck's medicinal chemistry innovation made this molecule Enlicitide 2003 Drug Hunter Molecule of the Year, because the amount of innovation almost make people wonder. It's a tour de force of the medicinal chemistry. It makes basically totally throw away the Lipinski rule of 5 and making this kind of micropeptide not only potent, also very good DMPK profile, so that you only need to take 1 pill once daily. So that's a very amazing story for Merck to pull this off. That's why no other company so far has even coming close to Merck in terms of having an oral PCSK9 inhibitor. So that's why it's the very interesting story. And then Merck's phase 3 trial is running very smoothly. They were able to design a 52-week only need 1 dose, 20 milligram daily dosing. And compared to placebo, it's a 2-to-1 randomization. And they also tiered, kind of stratified into those patients with high risk of ASCVD, cardiovascular disease, or relatively regular risk of ASCVD. So this trial is very well executed, and the primary endpoint is mainly the 24-week, the baseline, the percentage of reduction of LDL cholesterol, and they also look at 52-week as a secondary endpoint. And of course, the most intriguing part is lipoprotein. So that's something I will talk about later in the final slide, why lipoprotein A reduction by PCSK9 is a major advantage. Many physicians often may not pay enough attention to the reduction of lipoprotein A. That's a very important advantage of PCSK9 compared to statin. And of course, you also need to show it's safe in terms of adverse events. And overall, the data looks very impressive, as impressive as the antibodies. It achieved like 60%. Because Repasa, the antibody, is about 55% to 60%, so it's as good as the monoclonal antibody, but it's a daily pill. You don't need any injection. So overall, this 6% reduction is very impressive even at 24 weeks, and it's very durable. And then in terms of the stratification of diabetes or without diabetes, and they were able to show without diabetes, the percentage of LDL cholesterol reduction is similar at 24 weeks and compared to the other with diabetes. So very similar. And then they even dig a little bit deeper in terms of Merck's trial, also look at for those patients never had a cardiovascular event. The goal, American Heart Association recommendation is to get lower to 70 mg per deciliter. So that's a very interesting number because nowadays, in the statin age, people talking about 100 or 130 as the threshold. Now, American Heart Association really recommend 70. Of course, that's still debatable whether it's really needed, but that's definitely desirable. And for people already having a cardiovascular event, the American Heart Association recommend to go under 55 mg per deciliter. So that's Merck's oral drug achieve its kind of compared to placebo. It's interesting, the placebo still works 8 to 10% achieving that goal to get down to 70 or 55. But with the oral Enlicitide treatment, it's like 73 or 80%. It's significant, statistically very significant, both in patients without diabetes or in patients with diabetes. And then I really want to highlight as a secondary endpoint, it's not surprising that ApoB is also reduced because, as I mentioned, ApoB100 is the only apoprotein on LDL, whereas lipoprotein A is by genetics. There's so far no single drug is working just to lower LPA, and also lifestyle adjustment by diet or exercise won't move the needle in LPA. So in Amgen's case, they already published data. The Amgen PCSK9 antibody can lower lipoprotein A by 30%, which is pretty good. And Merck's oral drug works as well. So in patients without diabetes, the lipoprotein A reduction is about 28.5%. With diabetes, it's 27%. So this is huge. To me, that's because many physicians may not pay enough attention on this lipoprotein A, because this is another major risk factor. And so far, there's no drug. Maybe there are many drugs coming to reduce lipoprotein A. But right now, if you're taking PCSK9 drug, it not only lower the cholesterol, it's also lower lipoprotein A. So that I want people to really know why that's important. And in terms of safety, yeah, it's a very good safety profile. There's no major, it's almost similar to the compared to the placebo, like any adverse event. Treatment is 64%, placebo is 62%, and serious side effect, the placebo is a little bit higher. And in terms of deaths, yeah, it's also very comparable. So it's placebo-like and very good efficacy. So this will really potentially can be a game changer because the antibody PCSK9 always have trouble in the adoptance. Now that you have a similar to statin once daily pill, so of course the health economics will be the deciding factor to lower the price enough for the insurance, the payers to accept that. So now let me move quickly to the final topic.